Good evening, everyone. My name is Mansi Tripathi. I am TA for this course, Wastewater Treatment and Recycling. So, we are halfway through the course, half of the course is left. So, I hope you are studying well as you are keeping track of all the assignments. So, we are here to solve some assignment questions, not the assignment that you have got, some questions that are related to assignments. There will be some theory as well as few numericals. So it will be better if you take your calculators with you. I'll be sharing my screen, then we'll solve the assignment. So as you can see, we are in week 6. Hope you are going through the lectures properly. So this week we are talking about the treatment technologies as we were talking about in the previous week. So there are various treatment technologies that for water or wastewater treatment. So this module is specifically related to the wastewater, which is different from water treatment because it depends upon the end how much treatment we want to give in our wastewater depends upon the end use. If we want to make it drinkable, so we will treat it completely. If we want to discharge it into any land as well as any water sources, little less treatment will be required, but still we need to maintain some effluent standard so that it can be easily discarded, it can be easily discharged in the water body. Because in many cases we are using that water body as a drinking water source. So we'll be getting treated water, but some animals as well as some living organisms living inside that water source, they are also affected. So that's why we need to maintain some discharge standards. So I'll launch the polls where you can answer the question. I hope you will keep your calculators with you. So this is the poll for question number 1. You can find the polls in the right bottom corner of your screen. You can answer there. So the question is, which of the following statements are true regarding biological process in wastewater treatment? Biological process means we will use some biological means for treatment of wastewater. We are using less of chemicals or no chemical, but we will rely on the nature to treat the water or wastewater. So, which of the following is true? Removal of organic matter is achieved with the help of microbes. Microbe means small organisms, in short, biological means. Nutrients such as nitrogenous and phosphate rich compounds are also removed. Part of organic carbon is converted into gas, or all of the above. So, what can be the answer? Hope you can answer it in the polls or you can answer in the chat box as well. And in case of any doubt, I'll explain. You can ask in between. Okay, we'll look the answer. So the solution is D because we are doing wastewater treatment. So obviously we are removing organic carbon. Organic carbon is in the form of BOD. We are trying to remove the biological oxygen demand. So in the same process, we remove nutrient. There are some processes, not all the process can remove nutrients. Okay. There will be removal of nutrients in small amount in some processes, but there are various processes which are specifically designed to remove nutrients. So C will be there all the time. C is our target. This B, some of the organisms can do. And yeah, A, because this is a biological process, so obviously we'll be using microbes to remove this. So that's why the answer is all of the above. I'll close the polls for this one and launch poll for question number 2. So this is the question. Select the false statement from the following. Okay. So anabolism is an energy consuming process. Catabolism and anabolism are independent of each other. Catabolism is the process of transforming large organic molecules in small stable end products, and catabolism and anabolism occur simultaneously in the microbial cells. So there is one process, anabolism, other is catabolism. Both of them are metabolic process. Both of them are required for to obtain energy for cell growth as well as for the various life activities of the microorganisms. It's very like most of the people often get confused between catabolism and or anabolism. Not confused, they won't be able to remember what is catabolism and what is anabolism. They will interchange the definition. So this you have to you try to remember properly. Try to answer this question. Okay, we'll see the answer. Yes, correct option is B, that they are not independent of each other, they occur simultaneously. Now I will tell you a trick how you can remember it. Um, See, catabolism. There is one small word cat. This is a trick. If you have any better trick, you can share. So there is one word cat. What cats do, they will scratch things. They will make big objects. They will scratch it and they will scramble it. So it in what happens in catabolism, large large particles are broken down into small particles. That's what cat do. So what happens in catabolism? Large particles are broken down into small particles, and in this process energy is released. And what does when we talk about anabolism, there is an, so we can relate it to ants. What does ant do? They will collect small food particles and they will go to their home and they will collect it. So what happens in anabolism? Small particles are collected as a large particles, and in this process energy is consumed. So I hope the example of cat and ants will help you to remember this process. If you have any better, this is the way I try to remember it. So if you have any better option, then you can go for it. This is just a trick. Okay, we'll move towards next question. So this is question number 3. You have to match the following. This are option A, B, C, and this is second, and this is third. So heterotrophs, chemoautotrophs, and photoautotrophs. Try to answer this question. Yes, more answers. I got one answer. Okay, I got two answers. So these are terms that you need to remember. Okay. So first of all, we are talking about energy source. If energy sources always start from energy source. Okay, to remember such kind of thing. When we talk about energy source, so there are two sources of energy, either sunlight or chemical reaction. Whoever uses sunlight as energy source are phototrophs, and whoever uses chemical reaction as energy source are chemotrophs. That is one thing. Second, carbon source. Whoever is using inorganic carbon are autotrophs, and whoever is using organic carbon, inorganic carbon means carbon dioxide, okay, and organic carbons are heterotrophs. So first is heterotrophs. You can understand organic matter will be the carbon source. Second, chemoautotrophs. So chemo, what is there? Chemo means chemical reactions will be energy source and carbon source will be inorganic. Second is photoautotrophs. Sunlight will be energy source and carbon source will be inorganic carbon. So we'll see the answer. So answer is C. Heterotrophs are energy produced by oxidation of organic matter. Wherever we will see organic matter, so they are mostly heterotrophs because their carbon source is organic carbon as well as energy sources. So we can call them chemoheterotrophs also. Chemoheterotrophs, if we will say chemoheterotrophs, means energy source is chemical reaction, and hetero means organic carbon heterotrophs. So by this way you can remember, you won't forget it. Just remember the sequence. This is question number 5, question number 4. This one is memory based question. So you can answer this, and I am launching poll for this question. The required carbon oxygen demand, carbonaceous oxygen demand, nitrogen and phosphorus ratio for the growth of anaerobic. Important point is anaerobic microorganisms is. Try to answer this question. Anaerobic microorganisms, I hope you remember, they do not require oxygen to survive. Try to answer this question. We'll see the answer. So answer is A. See, they are anaerobic microorganisms, so they will be needing carbon source. Okay, and because they are anaerobic, they will use less amount of sunlight, so they are mostly heterotrophs. Most of the anaerobic microorganisms are heterotrophs, and heterotrophs require high amount of carbon. That is why COD should be high. Okay, that thing is very important. You should remember this. We'll move towards next question. Which of the following statement are not true regarding microbial growth phases in batch process? Okay, batch process and microbial growth phases and not true statement. Cell growth at an exponential rate is observed in the log phase. Stationary phases occurs when a nutrient depleted in the environment. Acclimatization of microbes occurs in the lag phase and rate of decay is almost equal to rate of growth in death phase. This is very important question. I'll draw the growth curve for you. These are various phases. Phase 1, phase 2, phase 3 and phase 4. This one is lag, this is log, this is stationary, and this one is death. Please try to answer this question. Which of the following statement is not true? Okay, I'll try to explain the concept. In any batch system, we are putting some nutrient, and we are putting some bacteria. What will happen? Since these bacteria are coming from some external environment, so they will take some time to acclimatize. So during this time is depicted by this lag phase. What will happen? In lag phase, small bacteria will become large. This is what happens in lag phase. The number of bacteria will remain same, but the size of bacteria will increase. Now coming towards log phase, then now these bacteria are aware what kind of environment they are living in. Now what happens in log phase? They will reproduce. The bigger cells will divide into two in binary fission case, and they will reproduce. So the size of the bacteria will be smaller than lag phase, but the number of bacteria will be more. That is very important. Here in lag phase size is less, in more, and log phase size is very less, smallest of all. Coming to stationary phase, the rate of growth is equal to rate of death. It doesn't mean they stopped growing. They are growing, but they are dying at the same pace. And here now food is less, everything is very less, so they will start dying in death phase. So here rate of death is more than rate of growth, or there will be no growth at all. Okay, so that is why in this, I'll clear the screen. So that is why option D is correct. Hmm? This is the question number 6, and these are the polls for question number 6. Which of the following is a valid assumption for activated sludge process, or in short ASP? ASP we call it in short. Steady state condition are maintained. The reaction that take place is both aeration tank and secondary clarifier. Aeration tank is partially mixed and influent biomass concentration is higher than effluent biomass concentration. Okay. So this is the question. Which of the following is a valid assumption? When we design an ASP process or activated sludge process, so what we do, we assume few things. We assume few conditions to make the calculations and design simpler. Okay. These assumptions may not be true, but we assume to make the design simpler. So what is the valid assumption here? Thank you. What happens in ASP process? There will be a primary sedimentation tank from where the water will go to aeration tank. Then the water will go to secondary sedimentation tank. So this is the ASP process. Okay. So let's look at the answer. So the answer is A. So one of the important assumption is that this process is completely mixed, stirred flow, completely mixed flow reactor. Okay, completely stirred tank reactor. Okay, so as you have studied this concept, what we have seen that steady state flow is there. Means what we were doing, we were this equation, we were equating to zero. Remember in previous lectures. That is why this process is in steady state condition. And second statement, we assume that bacterial actions are going on this in this, and in SST means secondary sedimentation tank, the flocks are just settling. No reaction is going on. That is very important. We assume that flocks are only settling. Only settlement is going on, no reaction. And aeration tank is partially mixed? No, not at all. We completely mix the aeration tank. Okay. Influent biomass concentration is higher? No, no, no. What we assume that influent biomass concentration is zero. We assume that no biomass is coming. Whatever biomass is there, it is generating inside this aeration tank. These are the assumptions. We'll move towards next question. Question number 7. The net rate of biomass growth is equal to, this is the same for the growth curve. We are talking about net rate. Net rate means growth minus death. Okay. From here to here, it's growing. After that, death is there. So what will be the net biomass growth? This is X and this is T. Okay, I received a response. This rate is what we call mu, not this rate. This is mu, this is S. S means substrate, means food. Okay, and this is mu. Mu versus S diagram looks like this. Ks is half saturation constant. Half saturation constant means this is S, this is mu max, okay, the highest value. Just imagine this is mu max, this is mu max, this is mu max by 2, this position. So whatever the Ks S concentration we have, we call it Ks. So this is the diagram that you need to remember. So the net biomass growth is equal to A, and this depicts growth and this depicts death. So net is growth minus death. That's how we do it. This is question number 8. In activated sludge process, oxidation is required for cell growth, oxidation of effluent organic matter, endogenous respiration of microbes, or all of the above. Try to answer this question. Okay, we'll see the answer. So the answer is D. In ASP, I told you now, all the functions are happening inside the aeration tank, nothing in secondary sedimentation tank. So here, we continuously stir it, we continuously mix it so that the oxygen from the air can be dissolved inside the wastewater. And what this oxygen do? It will help in cell growth. More the number of cell, more the microbes will eat up the organic matter. Okay, oxidation of influent organic matter. Yeah, whatever organic matter will come, we will try to oxidize it. Like carbon is coming, we will try to convert it to carbon dioxide so that it can go away. And carbon dioxide is not harmful. Okay, we try to do that. Whatever ammonia is coming, organic organic nitrogen is coming, we try to convert it into nitrate. So these are normal gas. These are the normal byproducts that are not harmful. So A, B. And how is C happening? Endogenous respiration means some oxygen is required so that more cells are growing. Okay, more cells are growing, oxygen with oxygen. What will happen? Food will be continuously going down. Number of cells will go up with the help of oxygen. Then cells, after some time, cells will start eating among themselves. So that is what we call endogenous respiration. Cells will stop consuming on organic matter because that is very less. Instead, what they will do, they will try to feed on themselves. So that will decrease the number of microbes also. When the microbes are reduced, the effluent quality will be very good. That is why all of the above. So this is question number 9 and I'll launch poll for it. Which of the following is not an advantage of trickling filter? Trickling filter, we have a box. This is mostly in round shape. Round will be there. It will be filled with aggregates or other media, plastic and all. We can fill it with anything. Broken plastic rocks, bricks. We can fill it with anything. So water will trickle down. So which of the following is not an advantage? Can be operated at the range of organic and hydraulic loading rate. Efficient ammonium oxygen takes place. There is no risk for clogging. Small land area is required for installation. Okay, thank you. I have received answers. So the answer is C. There is no risk of clogging. How? When we are filling it, of course there will be risk of clogging. After some time, the all the pores will be clogged. Okay, no water will be coming out, or the quality of water that is coming out will be very bad. That's when we clean the filters. So that's question number 9 for you. Now coming to question number 10. All sequencing batch reactors have 5 common steps as follows. sequencing batch reactor, we call SBR in short, because everything is happening in one system. Okay, in every other thing, what was happening in ASP? What was happening? Water was going to aeration tank, then the water was going to secondary sedimentation tank. But in ASP, all things are happening in the same batch reactor. What we do sometimes, we will divide it into compartments. Okay, so one thing is happening in this, other thing will happen in this. Water will go from here, will go from there, then go out. We do sometimes like this. So what is the sequence? Thank you. More answers? Okay. So yes, you are correct. The option is A. Fill, react, settle, draw, idle. Sorry for this disturbance. So what happens in fill? Water will be filled. Okay. What happens in react? We will switch on the aerator. So it will act like aeration tank in ASP. What happens in settle? We will remove these two things. Water we will leave as it is and it will settle down. What happens in draw? After some time, some things are settled down. Water we get here, good quality water we will get above. So we will draw it. We will not touch this part, but just we will remove this water. What happens in ideal? We will give it some rest time. We won't do anything. By the time it is ideal, we will use another time to fill. Okay. So we'll move towards question number 11. So question number 11 and 13 are numerical, so it will be better if you take out your calculators. There are no polls for it, so we will type the answer in the chat box. So the question is, an activated sludge treatment is to be designed for the treatment of 5 mld sewage having a BOD concentration of 160 milligram per liter. What is Q naught? Q incoming? It's 5 mld, which is equal to 5 into 10 ki power 3 liters per day. BOD concentration of 60 mg per liter. So S naught we will represent BOD means food or substrate as S naught. That will be 160 mg per liter. With the mean cell residence time. Mean cell residence time we represent by theta C. So mean cell resistance time is different from detention time. Okay. Detention time means for what time a liquid is coming inside and leaving. So this time is detention time that we represent by theta. What is mean cell residence time? A microbe is there, microorganisms. It is there inside. It will go out after some time. But from SST, what happens? Some things comes inside also, right? So this microbe, if it is going out, it will come inside also. So the time for which this microbe is inside the tank will increase. So that is why this thing is in days and our this theta is in hours. typically 6 hours. The effluent should have soluble BOD of 10 milligram per liter. SE should be 10 milligram per liter. Consider the following values. Y mean yield coefficient. What is yield coefficient? Number of biomass per unit food consumed. KD as we have seen in growth curve, this is degradation coefficient for these microbes. Return sludge concentration, MLSS concentration mean X. This is XU or XR or MLVSS. In this aeration tank problem, no, we always use MLVSS. We never use MLSS. So whenever the value is given in MLSS, we convert it into MLVSS. Typically, MLVSS is equal to 0.8 times of MLSS. MLSS means volatile, and this is means total, volatile plus fixed. So this will be taken as X, not this. I hope we have noted down all these things. I'll give you a few minutes to note. Please note down this also, because sometimes you may get confused. We always use MLVSS in ASP problem. Okay, we'll see the question now. So the reactor volume required in meter cube is?
Sorry, ma'am, I have just one doubt.
Yeah.
If you can go back to the previous slide.
Yeah.
MLVSS is 0.8 into MLSS.
Yeah.
If you
Oh,
say,
yeah.
sorry.
Yeah, we have some problem. We have some mistake in the question. We'll be using MLVSS now. So I'll rectify this when I'll upload the lecture.
Thank you.
Okay. Thank you for pointing it out. Because MLSS means fixed as well as volatile. MLVSS plus MLFSS. So that's why MLSS will be greater than MLVSS. So we'll see the question number 11. So the reactor volume required is? Here you have to give numerical value. That's why no options are here. We have some accepted range between which you can give the value. So if such kind of questions comes in examination, you can see the values can vary by, like, after decimal point they may vary, or 1 unit before decimal point they can vary. So there won't be any problem because we have a good acceptable range. So it's not like if you are answering it in 2 places of decimal or 3 places of decimal, it may go wrong. So no, that won't be the case. We have acceptable range. We'll see the solution. So you have to remember this formula, or as well as you can derive it, but derivation will take time. So it's better to remember this formula. So this is the volume of volume for aeration tank. This volume is for aeration tank. Okay. Reactor means aeration tank we are talking about, because as we have seen, it is the assumption that all the reaction takes place in aeration tank. None of the reaction takes place in secondary sedimentation tank. So we call aeration tank plus secondary sedimentation tank as ASP. But when we refer to reactor, we refer to aeration tank only, because no reaction takes place inside SST. So that's very important. So that's how we will calculate V. So please check this calculation. So as you can see, the acceptable range is very large. So there won't be any scope if you are putting the formula right, and even if you are using the round off value, there won't be any scope of mistake. We have good acceptable range, and this kind of range will be given in examination. We'll move towards next question. The volumetric loading rate in kg per meter cube per day is, this 5 is mistake. Answer. Loading, when we talk about loading, we talk about a substrate. In that respect, you can answer the question. I'll give you a few seconds, then we will see the answer. Okay, so this is the answer. Volumetric loading rate is Q S naught upon V. So it's written little far, so it will be like Q S naught upon V. What we have to do, we have to just convert S naught, which is given in milligrams per liter, to this range, kg per meter cube per day. So we can multiply by Q and divided by volume, we can get this value, because loading is calculated in the form of per mass per unit volume per day. So this is better value to understand. Actually, milligram per liters is there, but this value is better when we want to compare the performances of different secondary sedimentation of different treatment plants. When we give values in milligrams per liter, it cannot be compared in different treatment plants, because in some large plants, like in small plants, what happens, there is more fluctuations. In large plant, water is kind of stable, so that's why we cannot compare these two plants, because in small you will not get clear effluent because of fluctuation. So just to compare two different treatment plants, we put the value in this range, kg per meter cube per day. So it can help us to it can help us in ranking the treatment plant efficiencies. Moving towards the last question. The food to microorganisms ratio in kg of BOD per kg of biomass per day is, this you have to answer. Food is S and microorganisms are X, but take care of the units. Both the things are in milligrams per liter. You have to convert them into kg. Uh, like kg per day, we have to convert them. So this is we do this also just to compare the different efficiencies of the treatment plants. We'll see the answer. Same factor will be there, Q by V. See, as I have told you, we use only MLVSS concept, not MLSS, because only the volatile fraction, volatile means the organic fraction, will be used. Whatever there is fixed, it will remain there, it will go outside of the sedimentation tank, we will not use it. Not we, like microorganisms or the other things that are present inside the water, they will not use this thing. So this will be the food to biomass ratio, which we got 0.25, and as you can see that we have good range, a good acceptable range. So that's all for today. Any doubts?
Ma'am, can I ask a question?
Yeah,
Uh, can you just go back a few slides
yeah. Which slide? Question number?
where the question was?
Okay. This?
Yes, ma'am. The effluent should have a soluble BOD of 10 milligram per liter. Is this the final BOD?
Yeah, yeah. This is the final BOD.
Okay. Because I find this information not used in the
Yeah, yeah.
uh,
Yeah, information was not used in this question, but you will get question. Maybe I have given one question in this regard in assignment, the latest assignment.
okay, fine.
You will, in case of, if you want such kind of question, I can upload. I will attach one two questions more. Maybe
Thank
that
you.
will help. Yeah.
Thank you. Thank you.
Okay.
Thank you.
Thank you. Okay. So shall we close the session? Okay.
Thank you very
Thank
much.
you. Thanks for joining.